And welcome to the Land Grant Research Seminar Series today. Uh, Today is the presentation before last. So on next Thursday, a week from today, we will have the last presentation for this semester. It was a very productive semester. As you can see, I put a copy of the presentation that were given during the semester, title and also the speaker name. So to remind you of the topics that we had. So I'd like to, as I mentioned before, urge you to consider presenting, those of you who did not have a chance to present, and also to suggest some names. I'm very pleased to see that our postdoctoral fellows are really contributing. And remember the importance of postdoctoral fellow. You are the engine of research in any research institution. So I encourage you to really step up to the plate and present your work and get to know how to present your work and to promote what you have been doing and working on, because you never know. These presentations are online. Maybe you impress someone and they will hire you, or they will offer you a job, or get to know you. People get to know you through your work. So with that, I would like to encourage you, and I cannot emphasize more the importance of contributing to this. Also, if you see someone that is really important that we can, outside our campus, which we encourage to other to participate, to suggest their names, and maybe invite them, given the possibility of giving online right now. So I think that opens doors for us. But of course, we always prefer that it will be in person so that we can interact with that person, etc. For next week, we're going to have a team from Shell, 5 people who are going to be coming visiting us, and at the same time, there will be one of them, Dr. Andy Robertson. He will be talking about the nature-based solution in shells and their importance of research and development that they have been doing. With that, I would like to call on Dr. Aruna Urisuria to introduce our speaker. And our speaker, I get to be know she spent some time in University of Florida, and she spent some time, in fact, in the same building. I spent 9 years there, so it's very good to have some people from your home institution. So, Dr. Ursuria, please welcome our invited speaker today.
Thank you, Dr. Ferres. It's a pleasure to introduce today my colleague, Dr. Prabha Amar Singh. And Prabha received her bachelor's degree in agriculture technology and management from the University of Prayadhini, Sri Lanka, where I was also graduated. And then she received a master's in molecular life sciences from University of Colombo in Sri Lanka. Then she went to the University of Florida for her PhD degree. Prabha joined our research group last year, September, and since then she has been very productive, training students and moving towards the hemp cannabis research we initiated. Her research integrates genomics, cytology, anatomy, morphology, ecology of the plants using bioinformatics to generate predictive models, which can address both fundamental and applied questions. Um, so she's talking today about expanding horizons in industrial hemp, research in Prairie View A&M University, characterization of the hemp strains we have here, and potential applications of a byproduct. Please welcome Dr. Amar Singh.
Thank you, Dr. Veerasurya, for that nice introduction. And thank you all for being here today. I'm thrilled to have this opportunity to talk about what I have been doing here over
Yes.
the last 1+ years. And I want to give you an introduction about what I was doing before and my research area. Prior to join here, my research was primarily on conceptual aspects of plants. Specifically, I want to model evolutionary history of a plant group called Memicylon. It is commonly known as blue mist. And I wanted to use genomic data, internal structures, external structures, ecological data, and generate ecological and evolutionary models for this plant group. But it became a huge challenge because this plant group had no genomic resources before. So before beginning the research, I developed a bioinformatics pipeline to get molecular markers for this plant group, and this method is now used for many plants. After that, I collected plant samples across the old world tropics and used their genomic data, internal, external, structural data, and developed ecological and evolutionary niche models across the old world tropics, and found that how this plant group came to this world and how it dispersed and how their ancestors looked like millions of years ago. Thirdly, I collected samples from South Africa, these blue mist plants, and I found that there was an evolutionary reticulation within this group millions of years ago. Finally, I collected species occurrence data of Memicylon in Sri Lanka and modeled produced their niche models. And it explained that these plant groups lose their habitats or change their habitats as soon as 2050s. I got a lot of laboratory skills, then field experience and computational experience. And at the end of PhD, I was wondering, how should I apply this in practically useful thing? And I'm fortunate to get this opportunity to apply these skills to an amazing and versatile plant group, Hemp Cannabis Sativa. When we hear cannabis, an exciting thought comes to our mind is marijuana. Because the basic question is, are hemp and marijuana the same species or are they distinct species? Answer is no, they belong to one species. The key distinction can be explained using the phytochemistry. That is the THC or tetrahydrocannabinol percentage. This is the psychoactive chemical. If someone wants to dope, industrial hemp is not an option because it has less than 0.3% of THC by weight. Most of us know about THC. In 2000, we came familiar with CBD, that is cannabidiol. Cannabidiol helps calm your mood or keep your body relaxed without giving psychoactivity. But there are many more cannabinoids in hemp, also terpenes, which is responsible for fragrance. We get it from cannabis. This is a little bit about chemistry. I'm not going in detail to that, but before diving into hemp research I did, let's have an overview of some milestones in hemp industry.
Sorry about that.
Yes. Can I mute my mic? Okay.
You can mute them all.
Yeah, I did that, but you muted.
Okay, image slide notes. Okay, that's okay. Okay, sorry about that. I was talking about the milestone cannabis history, important milestones. In 1996, cannabis was first legalized as medicinal cannabis in California in the USA. And this is only under physician supervision. And with that, people were very optimistic and excited about these cannabis products, and green rush started with that. It is similar to gold rush in 19th century. Since 2012, 10 states legalized recreational cannabis consumption. In 2018, this hemp bill was passed, and it removed this hemp classification as a controlled substance. Instead, it was made as an ordinary agricultural commodity. And we are excited about Texas. We want to know what happened in Texas. In 2019, Texas legalized hemp-derived CBD products. By 2020, there are 3 states legalized medicinal cannabis. Still, this green rush is continuing in the USA. And to give you a bit of impression about the diverse products of industrial hemp, we have fabric, we have nutraceuticals, we have cosmeceuticals, there are cannabis cookies, cannabis milk, cannabis ice cream, and also cannabis vodka. So there are lots of products. But because of the industrial hemp has a lot of deficiencies in basic scientific data, our question is, are we navigating blind in the green rush? Because this hemp has a prolonged domestication history and a lot of illegal and legal hybridization, there's a huge genetic reticulation within the species. So it affects decision making. And also, there are unresolved naming issues of different cannabis strains. There are limited scientific methods to harness hemp byproducts during different productions. And there are limited research on stem rating in fiber extractions. To answer some of these questions, I worked on 5 major projects. And the importance of this project is there is an undergraduate involvement in all parts of these projects. Camille Perry involved in fiber strain characterization and understanding hemp rating methods. Asha worked on hemp rating methods, and she's also helping me with the hemp herbarium. And Anaya Ware, she is mainly working on grain type hemp characterization, and she's also helping me with hemp herbarium. And Robert Lewis, he's working on this innovative hemp wax project. Let's have a look on this fiber strain characterization and hemp rating methods, because these are interconnected and we did this project together. First, I want to give you an idea like how these hemp fibers have been used. It is not a recent commodity. We started using this hemp fiber thousands of years ago. This hemp fiber is used for fabric, ropes, fish nets, animal bedding, and paper products. In the modern hemp space, hemp fiber is used to produce coolers, activated carbons, batteries, sheets, auto parts, supercapacitors, and this cool hempcrete, which is an alternative to concrete, and also created hempy texture. So there are 2 kinds of fibers responsible for these hemp products. One is bast fiber and the other one is hurd fiber. If we take the stem of the hemp, it has this cortex which create bast fiber bundles in phloem tissues. And inner woody core has xylem tissues, and they have these hurd fibers. So this example of this bast fiber is the fiber we use for fabric, and one example of this hurd fiber is the fiber types we use for hempcrete. So how do we extract these fibers? That is using a method called retting. During hemp retting, we use different methods to dissolve the compounds or cells like pectin and hemicellulose compounds from these tissues and detach the fiber bundles. There are multiple methods of doing that. Chemical, mechanical, enzymatic, and microbial methods are prominent. So with this information, we wanted to test the mechanical properties of fibers linked to morphological and anatomy of hemp strains. And also we wanted to determine how retin methods impact mechanical properties of hemp fibers. Also we wanted to know and explore a natural method of hemp retting. There are several natural methods, but they have inconsistencies, but we want to come up with a new method. So initially we use 16 male plants. I have to emphasize here that hemp has male and female plants separately, and sometimes male and female parts are on the same plant. In this case, we use 16 potential fiber plants of non-fiber plants at the full flowering stage, that is approximately 90 days. And then we observed and measured macroscopic and microscopic characters. Also, we hand-sectioned the stems, different parts of the stems, and stained them and observed its microscopy and digitally measured them. We use all this data in statistical analyses. In retting, we use chemical, enzymatic, and microbial retting methods as outlined here. And then we use these fiber bundles and measured their tensile strength. Also, we measured the moisture retention specifically on hurd fibers. Then we did colorimetry of bast fibers. Finally, we use all these fibers and observe their internal structure, ultrastructure using scanning electron microscopy. We use all this data for statistical analyses. To summarize our results, this is the anatomy of stem parts that we got from middle part of the stem, that is the node 4 to 5. Here, red represent bast fibers, which is from the cortex area of the stem, and the blue represents hurd fibers, which is from xylem area of the stem. We didn't see any pattern of the middle layers. But when we tested the mature stem, they had a distinct pattern. Even in the replicates we used, we consistently saw these patterns. We wanted to check the bast to hurd ratio. We measured the cell layers digitally, and we didn't find any specific pattern in the ratio. This can be due to environmental conditions they respond to, and also their hormonal and genetic regulation. This is a point that we have to do gene expression studies. But we saw a link between tensile properties and this pattern in that hypocotyle area. For the bast dry weight, this x-axis represents our strains and the y-axis represents the average dry weight. And we saw that Bilobaski is consistently showing high bast dry weight in enzymatic, chemical, and microbial methods. Similarly, in hard dry weight, we saw a significant strain effect, but we didn't see any significant effect of retting methods. Here also, we recognize that Blue Genius is showing the highest dry weight. When we consider the tensile strength, both strains and retin affect tensile strength properties. And for moisture retention, we identified that strain effect was prominent. And there should be some other factors we have to consider, like surface properties of the tissues and the adhesion properties to understand how this moisture retention is controlled by the strains. When we observe them using the colorimetry, there are 3 color components, that is about lightness, chromaticity of yellowness, and chromaticity of greenness. And there was a significant effect of retting methods and strains. Yellowness was high in chemically retted fibers. Greenness is high in enzymatic and microbial retin, and this was consistent with the previous study. When we observe the ultrastructure, we noticed that microbially extracted hemp fibers, they gave like very well separated fiber bundles. Also, in some strain, we saw that there were like inconsistent diameter in the fibers, and in papers they were mentioned as dislocation, which affect the tensile strength. In herd fibers, one thing we noticed was that this cherry vine, which had lowest moisture retention, they had very hard, thick cell layers. We have to identify if this thickness affect the moisture retention. And when we input this data into PCA biplot, we understood that PC1 can contribute to more of the diversity, but we couldn't find any distinct clusters that can be due to number of strains we use and the characters we use. To improve that, we have to increase number of strains and the characters. With this, we can identify that we are towards our goal with these results. And we wanted to identify more about these microbial retting methods because accidentally we found that there is a bacteria in okra or abelmoschus, and we can use that for hemp retting. So we isolated the tissues which are infected with this bacteria, and we use biolog microstation to identify these bacteria. And we found that the most abundant bacteria was from Serratia species, which is responsible for rotting, soft rotting of fruits and vegetables, and that's a gram-negative and rot-shaped bacteria, and it has a potential capability of producing enzymes which can digest pectin, hemicellulose, and also lignin. When we re-inoculated this isolated bacteria again in healthy tissues of abelmoschus and hemp, we found that it's giving the same symptoms. We also wanted to know if this is a microbiome and what this microbiome constitutes of. So we did genomic extraction from these microorganisms, and now we are doing metagenomic profiling of these microorganisms. Still, this project is going on. So the next project is about hemp herbarium. Cannabis strains have unresolved nomenclatural issues. Their names are breeder reporter strain names. Look at the names they have. Most of them use cannabis language. Thousands of strains in the USA and thousands of names for them. So there are inconsistencies, lack of reproducibility in research, and there are identification errors. Also, it creates regulatory problems. We have to resolve this naming issue, and one potential solution is from herbarium specimens. I wanted to tell you what these herbarium specimens are. They are dried plant materials mounted on sheets. They can increase research reproducibility in plants, and they cause unambiguous reference of the results. So they are used in multiple applications these days. They are also used in omic sciences called herbariumics, and they are used in digital repositories like IOTIC Bio and GBF, and there are many more. We can apply this to herbarium specimens, and we established a new herbarium at the Prairie View A&M University, and now we have about 70 specimens of hemp. They constitute of wild, domesticated, and unknown strains. And we provided them unique identifiers using a barcode, and we are in the process of creating a digital platform for hemp. This became a very productive research with undergraduates. Anaya and Asha did hard work on preparing these specimens. And in grain-type hemp characterization, there's a huge diversity of the products of seed-type hemp or grain-type hemp, and there are huge diversity of the seed types, these strains. So we wanted to identify the morphological and anatomical diversity of this grain-type hemp, and also we wanted to understand genomic hotspots underlying the internal and external structure of grain-type hemp. For that, here we use 16 female plants. That is based on preliminary botanical data character sets we initially used. And then we harvested fruits and seeds and preserved them in a medium. And then we observed their microscopic and macroscopic characters, digitally measured them, and input them to statistical analyses. Also, we are doing genomic work on this. We extracted DNA successfully from 24 strains, including both male and female. And next week, we are sending them to genotyping by sequencing. Here, we are going to digest them using MSL1 region, and we will study the single nucleotide polymorphism of this. And the ultimate goal here is to use all this data and to generate models using machine learning, that is to identify the breeding values of each of these strains. To summarize the results, we identified that some characters like seed compactness. For example, in Jinma, you see seeds apart. In Sweet Sensi, you see seeds are clustered. So this is an important and significant character we identified. We found that ditchweed, among the strains we have, ditchweed has the most distant seed distance, and unknown strain that we found here, it had the highest compactness. And these are the 16 strains we use to identify characters of fruit characters and seed characters. We found 8 continuous characters out of 12 that are very significant here. And also we found an important distinct characters, that is the prominent trichomes. When we analyzed them by PZA, we found that these informative characters, that is discrete and continuous characters, both of them are like this. Here also we didn't identify any clusters, but they are almost forming clusters. That means we have to include more strains and more characters. The hemp wax project, here this is about a byproduct from CBD production. Depending on the facilities the companies have, they use either ethanol or supercritical carbon dioxide to extract CBD. During this procedure, there are multiple byproducts. Our focus here is this wax during ethanol wax suspension. That's the very first step of this CBD production. This wax showed lots of chemicals, hundreds of chemicals during this Francisco et al. Paper, and major classes included alkanes, terpenoids, and cannabinoids. They are rich with phytochemicals, and the question is, are we going to throw away this important byproduct? Then Dr. Aruna came with a great idea that we should test this on fruits and vegetables and see how it affects the post-harvest quality of this. Then Robert Lewis and I started working on this with the objectives of identifying a method to liquefy and solidify this hemp wax, and to find a method to apply this on the post-harvest products, and to test the ability of that to enhance shelf life of post-harvest products. We've tested different methods, different solvents, and to dissolve or melt the wax. And then we disinfected post-harvest products. Our initial focus was tomatoes and limes. In the experimental designs, we use fruits without coating as the negative control, fruits with commercial coating as the positive control, and 2 treatments. When considered tomato, in the treatment 1, we use unautoclaved wax as the treatment 1, autoclaved wax as the treatment 2. When considered lime, we use just autoclaved hemp wax. In the second treatment, we use autoclaved hemp wax plus a plasticizer called glycerol. This is a permitted food technology. We weigh these fruits every 24 hours, 9 days for lime and 14 days for tomatoes, depending on the perishability of them, and then imaged outer appearance every day and tested their inner appearance at the end of the experiment. So it showed that tomatoes without the coat ripen very fast. Compared to that, positive control and the other treatments, they showed delayed in ripening. And also it showed that there's a less rate of weight loss of this fruit after application of wax. When we treated this on lime, It showed that negative control, that is without the coating, there is a post-harvest disease on the peel, but in the other wax-coated fruits, there were no symptoms like that. And we identified that glycerol is acting as a very good plasticizer. In sensory perspective, this lime gave a nice fragrance because these terpenes have lime lemon-like smell. Although there are a lot of things we have to improve on this, our preliminary data showed that there is a potential of hemp wax to preserve post-harvest products. To summarize our research, during hemp fiber project, we identified a significant effect of hemp strain in all evaluated properties. And it showed that reti method affect hemp fiber properties like tensile strength and color. And there's a potential of using bacteria from okra to extract hemp fibers. And also we have a herbarium for the Prairie View A&M University. For grain-type hemp project, we identified a significant morphological diversity among seed-type hemp, and morphology contributed to a preliminary identification of grain hemp for genomics, which is going on now. And in the hemp wax project, we identified post-harvest products can be preserved using hemp wax. Still, there are a lot of work going on and many more to do, but we are confident that we are towards the goal. So let me wrap up here by saying that to secure the green rush, we have to use basic scientific data to identify the requirements of hemp as listed here and to improve the production and applications. If we do that, this green rush will continue and this hemp will become one of the strongest markets in the world. With that, I'd like to thank Dr. Aruna Veerasurya for giving me this wonderful opportunity and providing all facilities and conceptualizing multiple aspects of this project, collaborators, IT support, undergraduates, hemp, seed, and wax donors, financial supports, and CARP family being a source of strength to me during this research. Thank you so much for your attention, and I'd be happy to take some questions you may have.
Any question? We have some
Dr. Ferris, a non-technical question.
Okay, so thank you for the presentation. Just the result of the stress you put on it, right?
Yes.
Right, and so is that the strain at break of your fiber? So is that the result? So the stress that you put on the fiber, right?
Yes, it's a fiber flow.
Did it break when you stressed
Yes.
it? It did. Okay. So I was wondering about that, and then also you did some covering for the wax on the tomato and the lime. How can you be sure that when you autoclave it, because it's high pressure, right, high temperature, you get some degradation in your wax? How do you account for that?
Yes, that's a very good question. Actually, we noticed that even during autoclaving, it's giving like very strong smell.
Right.
And for that, actually, we designed two treatments with autoclaved wax and without autoclaved wax.
Okay.
They didn't give any significant difference.
Okay.
The reason of autoclaving is in a preliminary study, we identified after treating with wax, they perish very fast. To prevent that, we autoclaved it, and to identify the effect of autoclaving, we used unautoclaved and autoclaved wax. During that test, we identified there's no significant difference. In the second treatment, because no difference, we decided to use autoclaved wax because it's not giving any perishing effects. That's why.
Right. Okay. So.
But that's important that we have to identify the phytochemistry
Right.
of autoclaved and non-autoclaved wax.
Right,
Yeah,
right.
good question.
Right. Okay.
comes with infected plants.
Okay.
Usually the fungus like botrytis are there with the flower buds.
Okay.
So when you extract, the spores also goes to that, and then when you apply the same thing into the tissues, it will start growing. So the autoclaving reduce that, cut those contaminants,
Okay.
the microbial contaminants.
It reduced it.
Yeah, I mean, we can reduce means we can cut off the contaminants, microbial contaminants,
Okay.
live contaminants.
Okay.
The chemical wise, I don't think there is a huge difference. You will get the terpenes
Coming off?
yeah,
Okay.
out. But the main concern was to use this wax, just the waxy part
Right.
for the post harvest technology.
Okay. Just so preliminary. And so in the future, you will be measuring the thickness of the wax that's on the
Yeah,
the
but
product.
you know, the remaining terpenes and cannabinoids will help,
Yeah.
you know, in certain their characters also,
Right.
the help in the process.
Okay. All right. Thank you.
Thank you. To be fair to our online colleagues, Dr. Atai, you have a question?
Oh, a very non-technical question. Does CBD oil come from the fiber or does it come from the leaves? And the other part is that the fruit that you showed, are they about the size of lime? That large?
Okay. CBD basically coming from the inflorescence because inflorescence have bracts and these bracts have little hairs. We call them trichomes and they are glandular. These glandular hairs, the chemicals, cannabinoids are basically found on these glandular hairs. And in the inflorescence I showed, let me check my slides. Give me a minute. Oh, sorry. Okay. I think these are the inflorescence, infrutiscence I showed. Infrutiscence, there can be little trichomes because we found trichomes, but mainly they are not in this infrutiscence, they are in the inflorescence because inflorescence consists of little flowers and bracts. Mainly they have these cannabinoids. I don't know if I answered your question.
Okay. Thank you. We have a question there here from the audience.
Okay. Thank you, Dr. Prabha, for your nice presentation. First, I want to have a reflection on what Dr. Aruna said about the chemical composition after you just autoclave. You lose. I'm sure that we did a stability test of the wax compound. The wax, most of the monoterpenes will disappear after 65 degrees Celsius. You lose, but still autoclaving is good because there are some temperature-resistant bacteria and fungi. That's the nice approach. But I try to just say still you lose some of the important, like alpha pinene, beta pinene, all this will disappear. Okay, when I come to the question on the seed compactness, of, can you show us the previous slide?
Sure.
The one you showed recently.
Sure.
Yeah, this one. I can see the genema and the substances. They have clear morphological difference in terms of the seed arrangement. What it tells us, because you are an evolutionary scientist, as I can see from your introduction, what does it mean in terms of dispersal or survival, or is this because of domestication syndrome, this difference?
Okay, evolutionary advantage. One thing we have to find out, like when that came, like in the evolutionary history, is this seeds apart? That is an advantage, or seeds cluster is an advantage for plant itself. That is something we have to find depending on the evolutionary timeline of that. Maybe for your question, the question you asked, I hope that can be due to this domestication effects, because we wanted more and more seeds when we, like as industrial application, we wanted more and more seeds. I think selective breeding can be a reason, but that's a good point to find out.
Maybe the other point is one of your strain by below something. I'm not good in that because it is, I think, a Polish strain. Yeah. Originally, that strain is from Poland.
Yeah.
So, are you expecting any change in the fiber, the nature of the fiber, when they're just moving from one geographical location to another geographical location? Did you expect the same trend or different one?
Yeah, one thing I think is like for this fiber extraction, we take male plants because male plants grow taller. That is another evolutionary advantage, that they can pollinate. So genetically, they are tall, but when we change the geographical location, that height can be reduced, because in greenhouse condition, we notice that they are not growing that tall. They are growing short. So environmental conditions can affect that. That's also a good question.
Okay, thank you.
Great. Other questions or comment? We have one here.
Just to add a comment for the evolution issue, this plant has actually, due to the domestication and then restriction of the breeding by banning this for a long period and landing into the illegal growers, has created a huge problem for the natural evolutionary system. So Prabha actually didn't mention sometimes the plant is trying to survive itself. Now these are male and female plant. Sometimes when you stress the plant a little bit more, females started making male flowers, and the other way also. So that shows how much this plant has been stressed throughout this process and trying to survive.
I guess my question is, you make more product from you make more product, a better product from the male plant as opposed to the female?
Well, in the history, male plants were used for the fibers because male plants grows faster and taller. They want to put their energy to the maximums to grow taller, so that the wind pollinating plant can disperse the pollen easily. So in the history, male plants were used for the fiber extraction, females were used for the medicine. And in the beginning, in the very beginning, when Linnaeus found this plant in India, the THC content was very low, about 3%, right? And after this was banned and landed on illegal growers, they have bred this plant to the THC level today 40%, which is very, very high for even for the human brain. This is what they smoke now.
Wow. So are there plans to look at the properties of the male and female plant? I mean, the plant that has both male characteristics and female characteristics.
Without looking at the flowers, you can't differentiate male and female. But we are trying to get the genome data to differentiate male and female. That is obvious. And also, if we can if we can make the male plant sterile, then we can make more stronger fiber varieties, because after plant is using the energy towards the height and make more fibers to keep it upright, right? So after it started releasing pollen, the plant doesn't need to push too much energy for the fibers, right? So if we stop the flowering, we will get more strong fibers.
Great. Go ahead.
I didn't see the numbers on your single strand. I saw
Yeah.
the graph, but I couldn't see the numbers. I'm wondering how I mean, in terms of value, what it looks like.
Okay, tensile strength look like this. These are in megapascals, and these are for each strain, and the colors represent different rating methods.
Y-axis. What are the values in the y-axis there?
Okay, y-axis has tensile strength in megapascal.
The values.
Oh, values. Oh, values. They start from 200 to 800, and also 200 to 800.
Not quite as strong as steel, right?
Yeah.
But it depends on the thickness of the fiber.
Fiber, yes.
So it's because it depends on the thickness of the fiber. So it can be thinner, but it's a stronger compared to the same.
Also,
But anyway, it's an important properties, as you mentioned, for all those industrial purposes. So we have a question here for you.
Okay. Very good presentation. I enjoy that. A little high now still, so I'm going to ask a question. Hope it has nothing to do with me being high on the presentation. You work that you're presently doing, you're working with cannabis sativa, cannabis indica. Do you find any difference between those two strains?
Okay, well, right now I'm working on cannabis sativa, not cannabis indica, but in this project, like we are planning to have, because industrial hemp is cannabis sativa, but they have relatives, cannabis indica, cannabis rhodesiensis. So in this research, we will get some seeds in future.
To answer that question, actually, although we have cannabis sativa, indica, and rhodelaris before, the molecular data we use today show they are the same. So cannabis sativa is the same. That is why the confusion also. The marijuana also for cannabis sativa, industrial hemp also for
Thank you, Dr. Jung. Here he has a question.
Oh, yeah, it was a really interesting presentation. Yeah, just you did there's a 3 different experiment, right? It's almost a similar setup, but it's I have this a common question for the 3, but is can you go to that the setup for the data collection part? Any one of that case? Last one, first one, anything?
This one.
Yeah. Before that.
Before that.
Yeah, before. Yeah, like this setup. Yeah, each setup. Yeah, it's okay. Yeah, but this you mentioned it's a commonly used some ANOVA and PCA and Tukey, but it's you just show some of the outcome for the PCA only,
Yeah.
right?
Yeah. Yeah, ANOVA, Tukey, actually we provided only the significant levels here when.
Yeah, but this previously the kind of that ANOVA and multi, right? There currently is no outcome over there, right?
No, no,
Yeah.
it's not related to that.
Yeah, and also it's on the last one, last setup, you mentioned is the experimental design, but is you didn't mention any experimental design name.
Yeah,
And you
I
just
think-
that the component to which value you measure it.
Okay, I think it's this.
The last one, yeah, that one.
Yeah.
Yeah, that is you use the experimental design, and then is which component value will be measured like that.
Mm-hmm.
That is, you have to change. That is not experimental design, just experiment setup.
Mm-hmm.
Okay, experimental design terminology is not using over there. Okay, and one question commonly is, on this case, what is experimental design?
Okay, here we used 3 replicates from each, and we had this positive, negative, and control, and the experimental design we used was this is for one factor, single factor ANOVA. I think
Yeah, there is at least 2 factor on that page. Okay, first factor is that plus minus, that is the one factor, 2 different level. And the other one is you use the treatment, right?
Yeah.
Treatment 1. There is 2 different case. That is, can be is the second factor. Treatment 2 is a different, is a 3 different, 2 or 3 different level, right? 2 level, right?
2 levels.
Lime and tomato.
Yes, 2 levels.
There is a fruit type. That is the one factor. So this one is based on this information, there is at least 3 factor over there. Okay?
Okay.
So it's you didn't specify for your design, and then you just running for the PCA. The now is I'm not sure your PCA setup is right or not.
Okay. For this, we didn't run a PCA. This is just
That is mostly be based on your design. What I'm saying. Okay. At that case, your PCA outcome will be different. Okay.
Yeah. Yeah, that I will consider that. Thank you for that.
Thank you. And I think I was, before you may chip in, talking about the issue of dominance or the apical dominance, the growth of the plant, right? So yes, there is genetically, that's an important, but also you can enhance that through fertilization and also through light management by increasing nitrogen, so you increase the vegetative growth, and also by restricting the light, you push the plant to grow taller. So, and we have an expert on this domain, so maybe he can chip in on that.
Yeah, apical dominance is a complicated trait, so we'll talk about it at later time, okay? And if you have any question, you can ask me. But my question is, assuming that hemp is a domesticated plant, do we know its wild ancestor? And if it is known, how different it is from the wild ancestor?
Could you repeat the question? Sorry.
So assuming that hemp is a domesticated plant, do we know its wild ancestor? And if it is known, how different it is from the wild, so domesticated hemp?
Yeah, we have to identify that genetic distance of wild ancestors and domesticated.
Can you speak a little bit louder, please?
Sorry. We have to consider the genetic distance, like you said, about the wild relatives and the domesticated plants. But actually, I don't know which research is going on in that area. Probably Dr. Aruna has an answer to that.
Now, to look at the the wild ancestors and the relationship, you need to go for the original type specimens in Linnaeus collections. And I know they have done the sequencing on those plants. And once we completed our DNA extractions and analyzed, then we can compare with them. So the type specimens are there, collected by Linnaeus, right? So many hundreds years ago. Yes, yes. And so actually, there are some wild strains in US also. So we collected those, and what she referred as ditch weed is the wild ones. It may have brought from England, because English people have taken it from India to England and then England to here. And so it is not very clear, people were smuggling, bringing, yeah. It is tropical. It is tropical. That is why we can't grow it in the winter season here.
Great. I think one last question here.
Hello, I just want to say you had a really great and amazing presentation. I've had the honor just to work just with you on the project. I did want to ask you and both Dr. Aruna, what other hemp-based projects do you guys plan to work on in the future for the university? Like seeing the projects we've worked on, are there any possibilities for more potential projects going forward in food, preservation, or just anything else?
There are many projects actually. Dr. Kedani started working on extracted biomass to use in animal feeds, because we found those after the CBD extracted, the remaining biomass still has remaining a little bit of CBD and other terpenes that can be helpful for animal health in their cannabinoid system, and also to prevent certain diseases and parasites in their gut system. So as we move forward, we will find more interesting areas. In fact, one of the engineers in Tesla contacted me. They are interested using hemp fibers in their bioplastics. And the Toyota contacted me. They are interested using hemp fibers in their air filters. So these are potential things we can work on in the future with the engineering departments. And Dr. Ramre is planning to look at the carbon sequestration research in the spring, next spring.
Great. Well, thank you, everybody. Thank you for our speaker today, and let's give them a round of applause for the end, and thanks.
Thank you.